everybody, and welcome to our seventh Landcare Checks In for this year. If you haven't seen my face before, my name's Eve, and I'm the Member Services Manager here at WALN. I'd also like to welcome Vanessa, our Landcare Expertise Exchange Facilitator, and Georgia, our new intern who's joining us today from the WALN team. We are here to advocate, connect, support, and upskill the amazing network that is Community Landcare. But before we get started, I would like to acknowledge that I live and work on beautiful Whadjuk Noongar Boodja, and I consider myself fortunate to help restore this beautiful country. I'd like to pay my respects to elders past and present, and I extend a deep respect to all Aboriginal and Torres Strait Islanders on the call today. I acknowledge their ongoing connection to land, sea, and sky as the world's oldest continuous living culture. Please acknowledge what country you are on in the chat function. I'd love to see the diversity of land that we're all calling in from today. So thank you all for joining us online this morning. I'd like to welcome our speakers for today, Julie Fielder and Roz Hart, two Perth Fungi legends who will be familiar to many of you. They've done the rounds over the years, sharing their wealth of knowledge with us all about how we can best manage and support diverse and robust soil mycology. So without further ado, I'm going to hand over to Roz. Thank you so much for joining us today.
Thank you, Eve. So, Fungi4Land, we are delighted that the book has actually finally been produced and we can actually share it with you all and talk about the book. Yeah. So I too wish to acknowledge the country that we're on, and we, Julie and I, are on Whadjuk Noongar country, and we wish to acknowledge the elders past and present. It always was and always will be Aboriginal land. So the way this book came about is very interesting. It was not intended in the first place at all. Sapphire and I were actually doing a workshop down at Manjimup, and we'd done quite a number of workshops together, and we wanted this resource that we couldn't find. And we're going, where is this book about how fungi are important to our ecosystems and what goes on and why they're so integral? And after a while, searching and looking, we looked at each other and said, we're just going to have to write this book ourselves. But it did take us a lot longer than we'd intended. We found it was really important to gather and distill this huge amount of knowledge from Australia's leading experts on fungi, because we wanted this to be an Australian fungus book, not fungi from the other parts of the world, really on Australia. So it concentrates on how fungi impact our lives, our landscapes, our food, our economies, and how we impact on the fungi. That's important too. So we had a wonderful network of contributors, many people all over Australia, and we included New Zealand. We had some friends over there that we consulted with as well. This team comprised at least 50 people. So, and those who are involved with Sapphire and myself include Julie Fielder, who's presenting here with me today, and I'm going to mention some of the West Australian people. So Lynn Abbott, Anna Hopkins, and Mark Bondret. Of course, many of you know those people already. Our covers were designed a long time ago, and Anna Brady and Mars from Stray Orbit did a wonderful effect doing that. And then in the last year, when we realised we had so much to distill down, and we, as two scientists, were just so bogged down in our scientific content that Kevin Glidden turned our scientific content into what we wanted, plain English, and summarised each chapter with us and was a wonderful help to us. And Kevin is currently living in Colombia, so we were working between Melbourne, Perth and Colombia, which was shows what the modern world can do. Now, the basics that we look at in this book are what exactly are fungi and why do we need to understand them? And that is the integral part of this book. This is not a book about identifications. This is a book about what fungi are, what they mean to us and what we mean to them. So first of all, animals. Animals do a lot of interacting with fungi. Animals of all shapes and sizes, little animals, big animals, tiny insects, all have interactions with fungi, and many people are just totally unaware of this. Small digging marsupials like quenda are really important to fungus spore dispersion. And it's really lovely now. I was doing a fungi foray in Kings Park yesterday morning. We found quenda diggings, and the quendas are making such a difference to Kings Park. And they've moved into a lot of other areas in the western suburbs. And now we're teaching people, if you've got quendas in your backyard, it's a wonderful thing to have, and not going, oh no, they're eating my roses or something. So this is a real privilege when you've got quendas. So we have to change people's attitudes. The three-way relationship with quenda, fungi, and trees is so important, and it's really important for people to understand, and it's evolved over a very long time period. Insects also have very diverse relationships with fungi, and because it's also small, many people are not aware of what's going on. Some insects live inside fungi, some have specialist mouth parts because that's what they live on, just fungi. Others cultivate fungi in their nests for their food, and of course we have a really local example of this with the polyphagous shothole borers, which have a wonderful setup. They've evolved pits in the side of their bodies to shelter and carry their food-producing partner fungi, the fusarium, but they are causing us a huge problem, and they were brought in from overseas.
I probably need to tell the dog to sit down, but after the news of the last couple of days, I'm a little reticent.
Don't want to channel your inner Barnaby.
Yes, he's very happy to see both of us here. So talking about polyphagous shothole borer, biosecurity is our greatest protection against our biological threats. And we need to get everybody on board to understand we should work with the biosecurity, work with aquas, not regard them just as a nuisance at the airport. They are actually helping us. And coming back from New Zealand about 2 months ago, on the plane, the New Zealand biosecurity had the most fantastic video that they showed that I wish Australia would produce something similar about people who are affected by people bringing things in. And it was just the local person up the street talking about, please don't do this because this is what it does to me. And greater attention, funding and support and education of both all of those people here and also the regulators, because the regulators are making some terrible mistakes because they just are not biologists and they don't understand what's going on. Fungi can easily invade and they're very, very hard to control. So the best way is to stop them getting here in the first place. And as we found out, fungal outbreaks can have unexpected, awful cascading effects that people go, I didn't know that meant this. And that's what has happened. So people need to know that biosecurity is our protection and we need to all work with it. It's so important. One of the things I have found is in my local shop, in winter, they sell grapes that are brought in from the USA. Now, we as biologists know that a 1 in a billion chance of the wrong fungus coming in from the USA is going to decimate our grape industry. But they go, oh, but that's such a little percentage. We know it will happen. It will happen. So don't have any chance of it happening. So I found my son had grapes from the USA, and so I read him the Riot Act. And I have been known to stand next to the grapes from USA in the supermarket with people who are buying them saying, do you understand what this means? So I'm as mad as, yeah. We also have weedy fungi. So we don't just have weedy plants. And of course, weedy animals are called feral animals. We have weedy fungi, and they can cause a lot of harm to plants, animals, and humans. All we have to do is think about the court case that we had recently about beef Wellingtons, which are now going to have to be renamed, and the death cap mushrooms. So we don't have them in Western Australia yet, but we have oak trees in Manjimup. They will eventually arrive. And iNaturalist is going to be our really important platform so that we actually know when they arrive. And of course, the death cap comes from Europe. They're from other countries. We have a number of fungi now from other countries, and they damage both natural and built ecosystems. Okay. So conservation is so important. We who are in the conservation sort of sphere know how important it is, but many, many people have no idea. And our fungi conservation is even less adequate than the conservation of our plants and animals. We've got to include fungi conservation frameworks, and it matters to other species as well. It doesn't just matter to fungi. Things are not in isolation. Everything works together, and that's what biodiversity is all about. We have biodiversity harmony, where everything helps everything else. Many human activities present threats to our indigenous fungi in Australia, and lack of data and ecological understanding limits conservation. So we need more data, we need more information, and the best way at the moment is by teaching people to use iNaturalist. The climate crisis is real, it's here, it's happening, and threats to our ecosystem are arriving faster than anybody had anticipated. People need to understand fungi so that we can minimize human threats to them. And there are a lot of these threats. Some are as close and as simple as things that you do in your backyard. Some have easy solutions if people understand how fungi work. Just putting black plastic underneath the mulch that you then put on so that your local verge won't have weeds growing on it. Well, if the weeds can't grow down, the fungi can't grow up. And so fungi are being, confined to underneath there and they can't do their job. So the important thing for people to realize that fungi are underneath the ground all year round, doing their job of recycling or being partners all year round. So they're active all the time. And we need to take that into our brains as to understand what's going on. So when we want to put fungi to work, we would make a more sustainable future for all of us. Mycorrhizal inoculants have a lot of potential and need a lot more work. Each continent has its own mycorrhizal fungi, and we do not want to bring in mycorrhizal fungi from the USA or from Europe. Our landscape is very, very different. I don't need to tell you that. But people still think, oh, but in America, they could do this. And of course, the Americans are saying, buy our mycorrhizal fungi. They're really good. They're different to yours. Well, they don't work over here or they cause problems. So we brought in foxes. We brought in cane toads. We brought in things. Why should a different kingdom work any better? So no, do not bring fungi in from elsewhere. We always have some integrated fungi biocontrols, which are doing really well. We need more of these. And recycled fungi can be used to make biodegradable products that can replace plastics in certain areas. So that's something we really need to think about and work at. Now I'm going to pass over to Julie.
Okay, so we've got a chapter in the book where we're going to talk to you about a group of fungi called the recyclers or the saprotrophs. And these are really important for breaking apart the complex natural polymers we've got that mostly plants build. So that's things like lignins, which make wood strong. You can see the structure up the top. The cellulose, which is in leaves and wood, so about 33% of vegetable matter, and hemicelluloses, again, in leaves and wood, about 20 to 30% of vegetable matter. And then also chitin, which is made of the body of insects, but also the bodies of fungi are comprised of chitin. So if we want to, when things die and we want to have them recycled and those nutrients return to the ecosystem, basically, fungi are one of the only key groups of organisms that have the ability to break down these long, complex polymers that you can see up on the top left. They strip those little joining molecules apart. So very important. And they've actually kind of got their stomach on the outside. So fungal hyphae do this by they cover the surface of the material. And you can see some fungi on the left in microscope covering some wood. And they exude a whole bunch of digestive enzymes outside of their body onto that substrate. And then what they do is they absorb the nutrients back into their bodies from the substrate and mobilize them into the food web. So yeah, some nice little pictures down the bottom of scanning electron microscopy, having a look at cornstarch being developed. So you can see the lignin and then those little fungal hyphae running along and digesting it down. So in the book, you're also going to learn about biotrophs, which is fungi that join partnerships with living things. So we're going to introduce you to the concept of the meta-organism. Very few species exist in isolation without partner organisms, ourselves included, most of the animals included. So we have relationships with fungi and bacteria on our skin and in our gut to enable us to break down and digest food. Similarly, plants have these relationships with fungi as well. And so I've got some nice pictures on the right here of lobaria species lichens. We call these tripartite lichens because they have 3 different partner groups generally, sometimes even 4. So they'll generally have an algae to provide photosynthesis, a cyanobacteria that'll share sugars and nitrogens. There'll be yeasts, which is a type of fungi in the basidiomycetes, and also bacteria, and they improve drought and UV resistance and produce nutrients. So we've got this beautiful little synergy that goes on, this little symbiosis of several different organisms forming one species to be able to survive. So I want you, when you start to think about trees and plants, to come back to the lichens and start thinking about trees and shrubs being giant, more complex lichens. And so we have our arbuscular mycorrhizal fungi, which are the ones that form these little tree shapes with inside the root cells that you can see in the top left. This is one of Mark Brundrett's beautiful pictures from the book. The ectomycorrhizal, which is forms like a hearting net or a sheath around these roots, and they allow exchange. They grow in between the plant cells, and so they allow nutrient exchange to occur between the fungi in the soil and the tree. We've got our orchid mycorrhizal, so they have these little loops you can see in figure C. They're called pelotons, so they sort of form little hyphal coils that again are inside the plant cells, and that's what enabling that nutrient transfer between the orchid and its mycorrhizal fungal partner. And then the last one we've got is ericoid mycorrhizae, so our heath species, so things like Styphelia and leucopogon have these really special relationships. And they have hyphal coils that are extra fine and they fit inside the ericaceae very narrow root systems. So these are just some of the specialized mycorrhizae that we have. But every time you open a paper on mycorrhizae, you'll find people are discovering new types and new species and new groups and different ways that these biotrophs are interacting. So yeah, we're learning new things about these all the time, and it's really fascinating. It makes you understand that if we're going to be restoring vegetation, we need to make sure we have an understanding of these partnered organisms, these fungal organisms, because things like orchids, if we're doing orchid conservation, they won't reproduce without usually several partner fungi. So you'll have one that'll help with the germination, you'll have one that'll help them put up the seed leaf. And as you know, like a little tiny leaf, and then you produce this massive flower, that's because the fungi are often partnering with other species and channeling nutrient into that orchid to allow it to flower. So when we're thinking about conservation, that series of different orchid mycorrhizal that it develops over time is really important, and understanding that whole series is really important if we're going to look after our rare orchids. So restoration, we're still learning about fungal restoration, but there's this beautiful diagram that Kevin's made in the book to explain when we're moving from a dysfunctional to a functional ecosystem. And what we're trying to do is start returning diversity with all the different functional groups present within that ecosystem. So we want to make sure from a fungal perspective that we're getting our mycorrhizal fungal partners. So we've got our AMF, and then we've got our ectomycorrhizal fungi as well. And we want to make sure that we're developing the complexity of that over time. So some mycorrhizal will be quite generalist and partner with a lot of different plants, and we often find them in early stage recovery. But as we move up to a functioning ecosystem and high diversity, we want to be bringing in the more specific mycorrhizal partners. And so we're still exploring how to do that. So the issues we have with restoration ecology with fungi, we really know nothing about fungal dispersal biology at the moment. We don't understand how far spores go. We don't understand how often they germinate and actually produce like mycelium. And so there's a good paper that just came out a few weeks ago called 101 Questions on Fungal Dispersal Biology, where a bunch of the experts have gotten together and written 100 questions that sort of look at that research gap. So I would really love to encourage everyone to do some experimentation. So we've had a look at things like getting quendiscats or woolly scats that we find, and you can see the photo on the right-hand side here. We've got a huge diversity of fungal spores that you can find So this one's the one on the right. It's actually from a Gilbert's Potteroo project that Neil Bowger did down on the south coast. So we can also be relocating. If we find things like quendiscats in our native bushland and we want to be doing restoration projects, we could be collecting those because they've already collected a lot of different spores for us, watering them down and then spreading that through our restoration areas to try and bring those spore propagules back in to enable that connection to come about. Yep. So we've got a chapter here on soil. So fungi are involved in every level of soil formation, which we call pedogenesis. So they're really important for the weathering of parent rock. They exude enzymes similarly to when they're digesting lignin and actually can break apart the rock itself. They can also put their little hyphae in between rock sections and then pump them up with water and split bits of the rock apart physically as well. So I think it's something like 50% of all weathering of parent rock is fungally mediated. We also have biomineralization. So some sedimentary rocks form through fungi exuding things like calcium oxalate crystals through their bodies and creating actually sedimentary rocks. So sometimes when we look at them, they're created by fungi. As I said before, fungi are absolutely integral in nutrient cycling, being the only organisms that have the enzymatic pathways to break apart these complex polymers, like our lignin and our cellulose. They form stable soil carbon, and they often do this by exuding a chemical called glomalin that causes all the soil to stick together and gives us that good PED formation and that good water percolation that goes on as well. They also produce a lot of melanin in their bodies, and that's actually hard for fungi themselves to break down. So that melanin tends to remain in the soil in a stable soil carbon way. So super important for getting all the carbon into the soil. 90% of the carbon on Earth is in the soil. It's not in the trees, it's not in the atmosphere, it's in the soil. And so we need to be really focusing for climate change on getting that carbon storage of the soil back up. And I know Landcare does some great integrative work helping that happen. They're really important for nutrient exchange with plants, particularly things like phosphorus and nitrogens, and they exchange that with sugars with the plants. And they're really important for soil stabilization because their body, as you can see in the right-hand side of these long hyphal strands, they act as a very micro kind of soil binding, I guess they physically bind the soil together with it just by nature of their body shape. So one of my favorite things about fungi is it's a bit like an ecosystem. If you imagine being at the beach, if you imagine our soil horizons like being at the beach, and we've got the strand line, and then we move up the beach away, we get different habitats as we move through. It's the same when we dig a hole. So in that organic horizon, we've obviously got heaps of organic matter. We get our saprotrophic fungi, so there are recycler fungi in the top that are breaking that down. We get our small-scale generalist mycorrhizal fungi. We tend to get some pathogens and slime molds living in that top layer. And as we move down below the organic horizon into that A horizon, where there's heaps more minerals, phosphorus, we get lots more ectomycorrhizal fungi. We still get some of the arbuscular mycorrhizae, and we'll get wood saprotrophs, so things that break down buried wood and rotting roots in the soil. When we get down into the B horizon, where there's pretty much no organic matter left, we're just getting some saprotrophs and usually just large-scale ectomycorrhizal fungi, things like russulas and cortinarias, the bigger organisms. And when we get down to the bedrock, which is the group that we know the least about, obviously because it's right down deep and people don't generally look for life down there, we've got many poorly understood fungi, including our rock-inhabiting fungi that are undertaking that breakdown that we've been talking about. So we've got a nice chapter here on landscapes as well. And so we talk a bit about why preserving Australian forests is important for fungal diversity. So obviously our really wet forests, so things like the Karri and the Tingle down south, are real repositories for a large amount of ectomycorrhizal fungi, and you'll generally get a lot more diversity expression in what we call charismatic megamycota. So our big fungi that you can see with the naked eye, because a lot of fungi are invisible. They're so tiny you can't see them. And so only 17% of Australia is forested, with only about 8% of that 17% remaining as old growth forest. We've logged a lot of what's on hilltops, and that's accessible and flat. And it's about considering how we start to restore these landscapes as we move out of the wood chipping industry and use these lands for conservation. So we should be considering fungi and how we manage the forests, including in the regrowth forests. It's really important if we're going to make these healthy and resilient to climate change. So in Western Australia, we just got a new forest management plan where we're still undertaking thinning operations in, say, the 55,000 hectares of Karri regrowth that's going on. We really need to be having a think about whether that's worthwhile undertaking that thinning or not, and undertake some research to investigate that, because fungi don't like soil compaction. Timber harvesting machinery is really heavy on the soil compaction. So we'll start, if we crush the air out of the soil repeatedly, we'll have reduced particularly mycorrhizal fungi. So what we've seen in the thinning trial is there's a lot of saprotrophic fungi kicking in, digesting slash and wood post-harvesting, very few mycorrhizal fungi fruiting and expressing. So if we don't have enough mycorrhizal development, we're not going to have that resilience in the forest that we're going to need to preserve it through the changing and drying climate that we're experiencing in the Southwest Land Division. So another thing, we get fiery first responder fungi. So we talk about them a little bit in the book as well. When we have a fire go through, you generally have a succession of fungi that just occurs over at least the first 10 years before it starts to stabilise. So put up a nice picture here of Pyronema omphalodes. So this I took in Tassie at I had a long arm burnt dry forest site that was having real issues surviving because it was beyond the tolerable fire interval. And so we cool burnt it using with the fire service using some more Indigenous fire management practices. And you can see you've got that Pyronema omphalodes, and what that gets in, it gets in 2 weeks after fire, and anywhere where the fire's been a bit hotter, it gets in and stabilizes that ash beds as well. And it uses its hyphae to aggregate that loose soil, mobilize the nutrients that are in that ash bed, because obviously we get a large amount of mobile nutrients when we burn things, and it starts to feed those nutrients into recovering plants and seedlings. So really, really important. If you ever get a chance to go to somewhere that's been burnt within a couple of weeks, have a look where the hot spots are, and you'll probably see this little orange crust starting to form. So we've got another chapter where we're having a bit of a talk about the changes that are occurring. So obviously we're living in a ecological crisis point at the moment. And so we wanted to have a bit of a think about how the communities will respond to things like climate change. So what we found through the reading, and a lot of this research does come from other parts of the world. We haven't had a huge amount of focus in the Australian research sector, and it's definitely something that needs looking at and more funding, because as Roz said, our environment is quite different and a lot more drought prone than a lot of other places in the planet. So drought's starting to reduce species diversity, and it also slows down those that decomposition. You need a bit of moisture for those saprotrophs to really do their work, and when we're having long summers and super El Nino events, we're going to start slowing down that decomposition, which in turn is going to mean there's more fuels. We have increased temperature extremes, which means we get more fires, and we end up with a feedback effect between more fires, slower decomposition, increased fuel loads, more disaster situations. So yeah, really, fungi have a huge role in making sure we're keeping those fuel loads down over time. So temperature extremes tend to reduce fungal growth, reduce resilience, and reduce carbon sequestration into the soil. So fungi will, in dry, hot conditions, will switch from using their ectomycorrhizal partners and become more reliant on their arbuscular mycorrhizal partners. So we're seeing a switching in what, and the ectomycorrhiza are a lot bigger organisms, and are the things that really tie that forest together, big trees and shrubs. So they're becoming less reliant on their bigger organisms, and so then we're getting reduced resilience in our trees with those temperature extremes, and we found that shifting rainfall patterns tend to decrease spore production and reduce diversity at sites. So not great news there. So we've got a nice picture here for anyone who's coming to the seedling survival talk. This is the bushland of Piney Lakes around the Melville Eco Hub. And you can see here we've got the Burragoon Lake, which is a beautiful remnant wetland just across the road from Piney Lakes. And you can see there's fragmentation and urbanisation that's occurred there with Leach Highway running straight past the wetland. So if we've got fungi living in Burragoon Lake, it's very difficult for them to get under or over the highway to reconnect with the wetland of Piney Lake. So the smaller patches of habitat we have, generally it becomes an island biogeography kind of scenario. We end up with lower species diversity and urban remnants, lower genetic diversity because there's limited gene flow between sites. And again, we know very little about dispersal issues. Fungi need to meet in the soil and tie a little hyphal knot together to have babies. So it's not like a plant where pollen's getting transferred from one to the other. They need to meet, the hyphae need to meet and exchange genetic material. So yeah, and then if we've got too much space between with no habitat, we're going to limit that dispersal. Okay, so that's our talk for today. So yeah, if you're interested in getting a copy of Fungi4Land, which I really recommend, it's a great read. You can put it on your coffee table, you can read it to your kids. Roz has had great success trying to rope her grandchildren into collecting mushrooms with it. You can either get it from the website, you can get it from us at public events. You can also order it via university, so UWA Publishing on their website as well. And yeah, we'd be more than happy to email out the links for purchase if anyone's interested. So the other thing we'd really like to encourage you to do, because there's very little people that are employed as mycologists in Australia. Like even myself, I'm a self-employed biologist. 90% of my plants will be botanical because there's just not the work in the sector. So we're really reliant on the contributions of citizen scientists in Australia to understand what habitat these fungi are growing in, what the species diversity is, where they're growing. And so Fungi Map Australia has been running for about 25 years now. And so a couple of years ago, Roz and I set up the Fungi Map Australia iNaturalist project. So if you download iNaturalist to your phone, get yourself a sign up. You can just take some really great photos of fungi from your local area and post them up to iNaturalist. And if you don't know what they are, it doesn't matter. You can just put it's a fungus on it. We've got some people that do regularly check the site, and there's also a raft of people that volunteer their time to check these specimens and put names on them. It's a really easy way of starting to learn about the fungi in your area with help from experts via iNaturalist. But I will really ask you, if you're going to take photos, please take photos where you can see the top of the cap, but also the gills and the stems. If you're photographing mushrooms, you want to see as many parts of the fungi as possible. You don't have to get it all in one photo. You can post as many photos as you want with your observation. And you can also use, Roz and I often use hand mirrors to get underneath fungi because we're trying to not encourage people not to pull them up, particularly in urban remnants where you've got limited reproduction and that fragmentation issue happening. So really like to encourage everyone to get amongst that. And on that note, I'd really love to open it up for questions.
Fantastic. Thanks, guys. That was so blooming interesting, as per usual. Just wanted to very quickly add as well that we have a iNaturalist session that we did last year on our website and on our YouTube channel, which is really informative. And the video is also timestamped, so you can go in and just select which ones you want to watch, and you don't have to watch the entire session, basically. But yeah, if anyone's got any questions, straight off the bat, we can do that. Otherwise, we'll jump into the chat as well. Let me have a look.
I've got a question. Oh, sorry.
Yeah, go. No, go for it.
Someone else. I was really interested when you started talking about, collecting the poos from the little marsupials and things, and to help spread that fungi. I mentioned in the chat we should have some little small marsupials, but we do have a lot of roos. We've got a lot of cats and foxes around, unfortunately, but we do have a lot of beautiful orchids in some of the really untouched bushland on our property. But we're also trying to restore some areas that have just been cleared. They're poor sand, and we're trying to create corridors of Banksia woodland, and they're quite tricky, I understand, to get going. If we were to collect, if you were going and collecting like the poos from like a quenda, or even like in my case maybe a kangaroo, to try and transport some of that fungi, would you just soak it in water and then apply it like as a tea?
Yeah, so what we're suggesting at the moment is that you break it apart and sort of shake it up. So kangaroos, up to about 15% of their diet in season can be fungal. So you're not going to get the volume you get with, say, a quenda or a woylie, but you'll still get quite a lot of spores. So yeah, if you break it apart gently with your hands and then even just blend it or shake it up, because the fungal spores are usually 6 to 10 microns long, so they're really tiny, probably, and they're pretty tough. And then be spreading that through. Your Banksias aren't mycorrhizal though; they've got a different nutrient strategy for those poor sands, and they are difficult to restore. So that's really good. If you look at the Banksia Woodland Restoration Project, there's a raft of reports that Mark Brundret's put together. And they experimented with a lot of different things as well. Mark's obviously across his arbuscular mycorrhizal fungi being one of the global experts on it. But I'd have a look at that, because they did a lot of different experiments, and you'll be able to see what failed and what worked. But I think
Yeah, okay. I'm
it'll
trying
be
at the moment with diluted wormweed, soaking them beforehand, so we'll
yeah, I'd
see
be
how that
careful
goes.
because you've got that because they're adapted to low phosphorus soils. If you put too much nutrient in, you'll end
Yeah,
up with issues. But I think the big thing with the Banksia restoration is you need to get some cover happening, and things like Jacksonia, Furfuracea and stuff seem to pick up quite quickly on those sites.
yeah,
And then I go back in and replant with your Banksias after that, because working on
yeah,
that Banksia woodland restoration project, getting survival of Banksias was really difficult. But when you're planting on those harsh white sands and it gets super hot out there,
yeah.
I
I've
think
put in acacias and things as well.
getting some nursery cover in to nurse those Banksias back up will probably be your best bet. But
Yeah.
and obviously you get quite a lot of ericoid species and stuff within those communities as well. But yeah, your actual Banksias themselves are not mycorrhizal, so you don't
No,
need to worry
okay.
too hard about them.
Cool.
They've got big caterpillar-like roots that are particularly evolved for low nutrient soil to mine stuff out of them. So, but your other things like your Jacksonias and those other acacias and stuff will have both arbuscular and mycorrhizal fungi, and plus their rhizobium partners. So good idea for that for the rest of the community. But your Banksias itself, you're just going to have to expect a low survival rate because they're hard to get
Yeah,
into touch
we're
with and
planting a
search.
lot. Thank
Yeah,
you.
good luck. It's a good exciting project. Yeah.
Hi, I
Oh,
just wanted to add to Laura. You said you've got a lot of orchids on the sides. I've been growing orchids for a while, and they are very different mycorrhizals. A lot of them are decomposers, and I've worked in rain, like in bushland stuff, and I think the most effective thing, if you want orchids to most likely to come back up, is just to leave stuff as they are rather than mulching it. I've worked on some places where we left coastal tea trees as as guiding paths, and we actually found like green hoods flowering out of them. So yeah,
Oh, wow.
most of the orchid mycorrhizals are saprotrophs. Um, yeah, so just leaving your stuff. I know there's a method out there by Andrew Paget, and he's been experimenting with this for a while because a lot of the fungi will grow off like leaf mold. So people like will grab some of the leaf mulch and like mold it in a plastic bag and introduce it back into the potting mix. And he's actually seeing direct sowing methods onto this that works, but this raises a whole bunch of other concerns around pathogens and pests that might be spread around
Mm.
unintentionally. But yeah, in general, with orchids, just with the material, yeah, leave them.
Okay, thank you.
Thanks, Melvin. Also, just wanted to welcome any other co-contributors to the book who are here today. I think Sapphire's here and Melvin, I assume you are as well. Thanks so much for joining us as well. Um, Adam, did you have a question?
I do actually. Thanks. Thanks, Julie and Roz. Great little talk. I'm looking forward to getting the book. I work on Banksia woodland restoration, and biggest part of my job is weed control. So, unfortunately, we have to spray chemicals. We spray glyphosate, grass selectives, and geophyte selectives. So I'm really interested in minimizing the negative impacts of that, and interested if you know much about the interaction with fungi.
Look, we went down the rabbit hole on glyphosate while writing the book, obviously, because it's one of the more widely used herbicides. The thing with glyphosate is it tends to impact your arbuscular mycorrhizal fungi. So these are the ones that are generalist, will occur with all plants, including grasses, eucalypts, everything in between. And what it does is it drastically reduces the amount of hyphae that they produce. It reduces the amount of vesicles that they form. It reduces the amount of relationships that they form with plants, and it reduces the amount of spore-bearing structures. So Basically, it's drastically reducing the fitness. And studies I've read on ectomycorrhizal fungi, I think because they're larger organisms, they're not as impacted as the arbuscular mycorrhizal fungi. But the thing is, so plants code for the Shikimate pathway, I think, twice genetically, and fungi code for it 16 times in their genome. So they're a lot more susceptible than plants to the effects of glyphosate. So, look, I tend to take a fairly pragmatic view of it, but I think having enough budget. My housemate works in bushland restoration, and I know there's been projects where if people are not given adequate budget to undertake really selective weed control, then you run into problems with people not having the time to really look at what they're spraying. And I think, things like Hart or Calisina, you're very limited with other control methods. But I think just making sure there's enough budget, making sure that your staff on the ground have a really good understanding of what they're spraying and why, because I think a lot of people get sent out without good grass identification. And, using mechanical control as much as possible would be the way I do it, but it's not always practicable from a budget and logistics standpoint. So I think realizing that a lot of these herbicides have large effects on the fungal microbiome. We're still learning about what's even there, let alone how it affects it. But the initial stage is that even things like aspergillus are being affected at 100th the scale of dilution that's recommended on label, and that it requires further investigation. And look, I'm hoping over the next year that we can sort of rabbit hole a bit more on different chemicals that people are using. And Adam, I'd love to have, I'm coming into the city of Coburn in a couple of weeks because I'm doing some contract work for you guys. It'd be really great to have a chat about what you guys are using and get a better understanding of what's being used, so we can go and do some more reading and come back with some answers on other pesticides and herbicides that are being used and what those impacts are in bushland management.
Thanks a
Yeah, and
lot.
that's a great question, because one of the things that we're aiming to do with the book is get people do a lot more research and ask a lot more questions and say, why is this happening? What's going on here? What if we do this? What if we do that? So that we share what we've actually found out. So this is really a starting point, so that we have some of the knowledge that we can actually then go out and look at things and find out what's going on and go, oh my golly, I had no idea that was going to happen. So it's really important that we get out there and do a lot more questioning and research.
Oh, it's an incredible resource. And just want to quickly jump into the chat. Mike Norman, do you want to just jump on and just ask your questions instead of me reading?
Yes. Can you hear me?
Yeah.
Yeah.
So one of my questions was similar to the question or the comment that was made that my bit of Banksia bushland today is being sprayed with grass selective fusilade. The reason being that we're having trouble controlling the annual veldgrass. We've got the perennial veldgrass pretty much under control now by cutting them out at the base, but the annual. So I was concerned of the impact that would have on the fungal associations and the prevalence of the right kind of fungi. So that was and that was answered a moment ago. And the other one is I'm about to plant some Christmas trees into that bushland, some WA Christmas trees, and I wondered if that might, because they're not there currently, although they do occur in the area, whether they would require to grow some specific mycorrhizal fungi. Otherwise, I might not be able to successfully get them growing.
They will often form, obviously, because they're the largest hemiparasite on the planet. I think you just want to have some healthy, robust, already standing trees before you put those in. But I think replanting them is a great idea. But yeah, I haven't specifically looked into the muja and what grows with it, and I'm not sure anybody has, to be honest. But they tend to be really resilient. Like when you end up with quite degraded areas, they tend to hang on a really long time. They're usually the last thing left when you're moving down that degradation because of their ability to parasitize other plants, and they can be quite virulent. So I guess making sure that the plants they're going to parasitize have enough vigor before you pop them in would be my concern. And then I reckon they'll be the last, if it all degraded again, they'll be the last thing left standing. So yeah, I
Right.
reckon it's worth a go. It's a good question though. I really like it.
Yeah. And the other one was just a comment that I run a coast care project over 2.5 km of coastline. We are manually removing most of the weed species. And by getting them out before seed set or shed, we've already eliminated 13 weed species totally.
Oh,
No
that's
longer
awesome.
occur on the site
That's
at
great
all.
to hear. Yeah.
And but it's taken 25 years. And many of
You've
the
got to be
others,
determined and persistent.
many of the others are now in low density. So, and we're
Right.
aiming to remove some of the others. So I think we're the first coast care group that has totally eliminated seaweed and marram grass from the
Oh, that's
dunes.
amazing.
Amazing work,
Excellent.
Mike.
We've
Where's
almost,
where's your
sorry,
project? Whereabouts is your
it's in
project?
Sorrento and Marmion on the west coast
Oh,
here.
fabulous. Yep.
And as a result of the intensive weeding, we've had a lot of natural regen. The only thing that has knocked back the regen is two things: trampling by beachgoers and rabbits. So we're trying to deal with the rabbits because one rabbit can clear out a hectare of natural regen. So rabbits have been a problem.
Yeah,
I feel you on the
absolutely.
trampling. We're having the same problem down here in Fremantle. I think lots of people, we have a lot of international tourists and they're just not brought up to not walk in the dunes like we are. And I think definitely there needs to be more education around the fragility of dune vegetation and destabilisation, especially with sea level rise and climate change. So I feel your
Yeah.
pain. We're definitely losing stuff down here in City Coburn.
Yeah, some of the trampling is due to a homeless person who lives under
Yeah,
a cliff, and
yeah.
you know he walks down there, and we said, well, at least can you stay to the one path?
Yeah.
Yeah, yeah, it's got a
Cool.
lot
I'm
of
just going to
fault
head back to the
going on.
chat as well. We've got a question from Scott, who works in environmental restoration. He's particularly interested in fungal ecological restoration and asked, are there any species that are exceptional for recreating fungal health in native regeneration? Scott, if you want to add any more detail to that, please feel free.
Yeah, plants particularly, because obviously plants are something that I can put back into the landscape. But that's what I was wondering, if there was any plants in particular that were sort of forerunners of the restoration process.
Good
Um,
question.
I think there's a couple of ways around this. You can either be replanting your climax species as well, or you can plant successionally. What we find with fungal restoration, because we're giving a talk obviously in a week and a half on the seedling survival. So I've been sort of having a bit more of a think about what that succession is. I think early on in your restoration, you just really want to be getting some ectomycorrhizal generalists and things like that back in. So things like Piezolythus and Leucaria, things that would naturally start to come back in over time. And then as your restoration is growing, really looking at like importing scats and stuff as an inoculum process. There is some experimentation going on with sampling soil cores from adjacent vegetation and then placing the intact soil cores in your restoration. My feeling at the moment is this is really early stage, so I don't want to definitively say whether it works or not, because obviously soil is a very diverse, it's very diverse and it is very site specific, both on a broad scale and also on a really fine scale. We find there's a lot of fine scale partitioning that occurs in soil fungi, but look, I don't think it's going to do any harm taking soil cores from your adjacent bushland and moving them over. It's just as to whether that's going to be successful, but I think I really think that's something that would be worth looking at using high throughput DNA sequencing techniques and stuff to see what's surviving over time and what's taking when we're transplanting between similar soils. But I think if you're going to look at soil core like movement, you really want to be matching up your plant species to what you're replanting. You want to be matching up your soil type, and also like if you're taking stuff out of the top horizon, you want to be getting as much of the horizon as different horizons as possible, because we have those different ecosystems occurring as we move through the soil profile. But I think there's a lot of unknowns at this point, insofar as how we're implementing that into restoration. And I think just getting people more aware of it, and then, trying to push for getting more university and research funding to look at these. So I think having people that are actively engaged in restoration, like so many people in this forum are, linking up with people who've got access to high throughput DNA sequencing and asking questions when people are doing funding. Because obviously, we've started looking at climate adaptive plantings and moving species from further away to cope with the climate change shift. I think similar things are going to happen, but we're still in our infancy in understanding what fungi we've got. 80% of them are still undescribed. When you do a high throughput DNA sequencing, you'll get fungi back. You won't even know what phylum they're in. So that's that level below kingdom, that first group. There's still unknown things, and we're discovering new ways in which fungi relate with plants and ecosystems all of the time. But it is a really underfunded portion of research. So part of bringing this book out is to give people that base knowledge, so that when we come in and we've done some, learnt new things and we want to share them, we're not having to work from the building blocks, because everyone's already read the book and got that back funding. So I'd love to give you a nice, tidy, definitive answer, but the other thing about working with fungi is it's rarely tidy or definitive. Everything breaks the rules. So yeah, but I'm really loving all the questions people have. It'll give us a good frame to move forward with, teaching ourselves more that we can then share with people. Because as I said before, a lot of this work, like, I probably spent 5 or 6 months reading just full time on top of an honours degree, just to pull all this stuff together, and I don't get paid for that, and I still need to eat and pay bills. So it's great to have targeted questions that people are actually want to know, that we can share, that's got practical applications for people who are on ground in the restoration sector. So yeah, feel free to email myself or Eve any questions, any of these sort of things you're interested in, and we'll try and keep building that knowledge base. So we can share it with everyone.
Agreed. Do we have any more questions from the floor? Ness, I saw your question about, I guess, how fungi recover post-fire, but I imagine that's probably a bit of a Pandora's box, isn't it? It's just like, you could talk about that for hours, I imagine.
Probably.
Yeah. I mean, look, there's some really interesting ones. We get some fungi that live in the old large logs, and they put sclerotia. They build a big ball in the soil, and then when the log burns out, that big ball just fruits 2 weeks after and chucks spores everywhere. So they've got really interesting strategy. Obviously, when you have a fire, the first plants that start to establish and cover the ground are things like our fire response mosses, like Funaria hygrometrica. So a lot of things like Anthracobium muelleri that form mycorrhizal relationships with mosses will fruit in abundance within the first few months following fire, after you get a good dump of rain. And then we start moving through things like foliotas and Tephrocypes that start coming back. And then till we're building up to that more climax community. But you'll still be picking up new species, and the succession will still be rolling from the study we did in Carry Forest after 10 years. And then in long arm burnt old growth forest, you'll get a whole nother suite of fungi again. Generally, your diversity will be a bit reduced, like it is with plants, but you'll have this climax, and then it'll create really good fauna habitat. So, fairly similarly with most things, we want to be making sure that we're sitting within those tolerable fire intervals, so the fires aren't too close together. We want to have some stuff that's long arm burnt, but we don't want to leave all of it in a really long arm burnt condition like they've done with the private land conservation estate in Tassie, where you start losing all that diversity and you're outliving the ability for things to reproduce. Also, if you end up with huge amounts of fuel, it doesn't seem to sporulate quite as much. You don't get as much of fruit body production in those long arm burnt systems as you might get ones. So
all makes sense. Thanks, Sapphire.
I've
I'm also going to collate all of the resources that Sapphire and other participants have very kindly posted in the chat, and I'll send all those out afterwards. So if you've missed those, I'll get them out in our post-event resources.
I've got a little comment, if that's okay.
Go on.
So Well, I live in Suburbia on it was originally a cleared block for the last 15 years or so. And I guess so I planted a heap of natives, and I've had the odd bandicoot bouncing around. And last year before, I actually found a native orchid totally randomly in the middle of my garden. And the only way I know that it would have got there was probably through the bandicoot poo. So I
Awesome.
love that.
That's lovely to hear,
So
isn't
cool.
it? That's great. You're
And this
doing
is
the
in the
right
middle of
thing.
Suburbia.
Yeah.
Yeah, so cool.
Yeah, nature's resilient. If you create the environment for her to come back to, she will. And I think that's the big take home. You know, it's a bit, if you build it, they will come. And I think so making sure that you've got, you know, the different components, like different types of coarse woody debris, you're minimizing your pesticide use, you've planted the plant partners, and you're trying to encourage places for dispersal vectors to come back in, so insects and you know, the cute and furry animals, then we've got a chance of building that resilience over time.
It's awesome.
Amazing. Yeah, as Julie and Roz have alluded to, they'll also be presenting next week at
Yes.
our Boosting Seedling Survival session at Melville Eco Hub, which is actually sold out in person, but you're still welcome to register for online viewing. That's going to be amazing. And from what Julie's said to me and Roz, it's going to be full and interesting. I'm really excited to have a good deep dive into all of the different elements that we're going to look at. We're going to look at root structure. We're going to look at nursery best practice. We're going to look at fungal succession. It's going to be amazing. And yeah, we're so excited for that one. And also, if we don't have any more questions, I guess we'll just kind of wrap up the session. It's been absolutely fascinating as always. Thank you so much, Julie and Roz, for sharing your information with us.
Thanks everyone for coming.
Yeah.
Yes, Laura, just quickly, we will record. If you register for the session, you can have access to the recording afterwards. But yeah, in the link to, in the post-event resources, if you're not already a member of WALN, we'd love you to join our collective voice for land care. I'll post a link to our humanities, which has kind of got all of our upcoming events. We've got a really awesome session with Dr. Kit Prendergast coming up, and we're going to be talking about native bees and planting for native bees, as well as funding sources available for individuals and community groups to access funding for native bee surveys. And we also excitingly announced our annual gathering in 2J at the end of August. That's going to be fantastic. We're going to be doing some wildflower walks. We're going to be putting a spotlight on all of the amazing work being done in that Avon catchment. And it's going to be in the beautiful Memorial Hall in 2J, which is like an 1800s heritage hall. It's absolutely beautiful. So really excited for that one too. And just wanted to again thank you all for spending your morning with us. And have a lovely day. Thank you.
Thank you.
Thanks, everyone.
Thanks. Bye.
Bye.
Bye. Book for 2J.
Definitely.